I will be talking to you about omics signatures of cellular perturbations, and then how they fit in this adverse outcome pathway concept, and then how you could actually use them using the portal that we developed named iLinks to actually analyze omics data using these signatures in the context of adverse outcome pathways. So, okay, so let me start with the signature, right? So this signature concept is really pervasive in omics world, right? It's everybody's developed some kind of signature, and they can consist of very different types of information, right? So I went back to the actual basic definition of what a signature is, what kind of characteristics anything has to have in order to be called a signature. And actually there is a biology and medicine centric definition of a signature, and it states that it's a distinctive characteristic or a set of characteristics by which a biological structure or medical condition is recognized. So in our world, these characteristics are some kind of readouts, molecular readouts of biological system. And then the key feature of whatever we call a signature is that it's discriminative, right? That it's kind of specific to something, right? In this case, biological structure or biological state in our context more often. So I will be talking about signatures of perturbations. And what does this really mean? Well, the perturbation can be anything, right? So it can be environmental exposure, disease can be a perturbation, drug treatment can be a perturbation. So any kind of perturbation of a biological system, and then we try to identify a signature that is specific to this particular perturbation. So anyway, pretty much like 80% of omics experiments fit in this description of perturbation. And if anything, so they can be kind of acute type of perturbations, right? Treating something with a biological system with a chemical, or it could be a type of chronic perturbation that eventually leads to disease, or it is a disease itself. So and then the signature of such a perturbation or effects of such perturbation of biological systems is basically consists of a pattern of changes in gene or protein expression levels after a perturbation. So I start off with this general omics world, right, that encompasses other things like metalomics and other omics. But really what we are talking about in this talk are gene and protein expression levels, and even more specifically, I will be speaking most of the time about gene expression levels, although iLinks portal does have protein expression data, proteomics data, and the signatures derived from such data sets. So at the end of the day, our transcriptomic or proteomic signature of a perturbation will be a set of all genes or proteins whose expression was modified by a perturbation, and perhaps some kind of quantitative measure of the intensity of such a change. And again, when we build this signature, we will try to build it in such a way that makes it capable to discriminate between different perturbations. Now I'm going to kind of switch to this adverse outcome pathway concept and what it really means, right? I picked up from NTP. And it's defined as a basically conceptual model that identifies the sequence of molecular and cellular events required to produce a toxic effect when an organism is exposed to a substance. And if you look at there is a little diagram there as well that describes what exactly we mean by that. So basically this model encompasses the initial molecular events that basically by which a toxicant interacts with the biological system or a living organism, right? Which could be some kind of interaction with a protein, stimulating a receptor, binding to a DNA, causing protein oxidation, or anything of that sort. This initial event then initiates the cascade of responses in the biological system that kind of can lead to activation of different genes, production of corresponding proteins, altered signaling, cell-cell interaction, and so on. I would, in this, if we are thinking of these in some kind of order, this list, I would probably put altered signaling on top, and then would follow by gene activation and protein production, right? Because any of these events will kind of trigger some signaling cascade that will eventually lead to gene activation. And then these changes in the proteome ultimately will change the functioning of the tissue and can cause changes in tissue development or function, ultimately leading to an adverse outcome, which is a disease or impaired development or reproduction. So how do we use omic signature in this context, right? So we will basically use signatures of different perturbations that we know what they kind of do in some sense, either which part of the biological system they are perturbing or what kind of adverse outcome they are leading to. And we're going to use them to basically compare to the signature of the toxicant for which we're trying to construct adverse outcome pathway, and basically try to list all these molecular initiation events and then subsequent changes in the functioning of the cell, all the way to the actual adverse outcome for a toxicant for which we are given a perturbation signature. And in this sense, we can sometimes be looking for figuring out what is the mechanism of action of a toxicant for which we know ultimate adverse outcome, or we can try to figure out everything, right? Have some kind of perturbation signature for a toxicant, and then try to figure out the mechanism of action, which I would say maybe constitutes of these first two boxes, and then ultimately also the potential adverse outcome. So the data that I will be using and came from this project. Not quite sure if you heard by it by now. It's becoming more known, but it's still somewhat obscure probably. It is NIH Links program, which was a big consortium aimed at creating network-based understanding of biology and so on, right? But the point is that the objective of the project was to generate perturbation signatures on a large scale using different assays, querying different compartments, right? Creating proteomics, transcriptomics, metabolomics, and other signatures. And for a large number of varying perturbations, not just perturbing agents, but also diseases and so on. So the concept of the links, not I-links. So links actually came about from this concept of connectivity map, which was introduced in 2006 in this paper. And the idea there is basically that we can identify putative therapeutics for a disease by comparing transcriptional signatures of the disease to transcriptional signatures of drugs, and finding drugs that are with negatively correlated signatures, which then would imply the drugs are perhaps capable of reversing the disease process. So the way this would work, right, you have your disease signature that can be constructed by either comparing disease and non-disease tissue, for example, in humans or in some kind of model. And you have reference database of signatures that are constructed by treating a cell line with a specific drug and then observing changes in gene expression. So all of this is basically transcriptional. So these dots here would represent upregulated and downregulated genes in the disease signatures. Here we would have the same genes, what happens to them in the signatures of a drug, and we would be looking for situations like this in which genes that are downregulated by the disease are upregulated by the drug, and vice versa, and that would give us then potentially therapeutic agent that could address the disease. So it's a very simple concept. It sometimes works. Most of the times it doesn't work because it's probably oversimplified. But in many situations offers a very valuable insights, even if it doesn't result in figuring out the drug or treatment for a disease. And so links represents a scale up of this idea, right, where we went from a few thousand of transcriptional signatures to millions of transcriptional signatures, from 1,000 of chemical compounds, here drugs mostly, to tens of thousands of chemicals, and then include what are added are disease and also signatures of genetic perturbations of genes. So just briefly to give you an overview of the scale of these, there were 6 data generation centers scattered around the country that were generating this links cube, which is basically combination of cell types treated by different perturbagens, again in a wide sense of this word, and across different assays. So the cube is sparse, meaning that for some assays, for example, we could construct a complete slice, right? For example, for transcriptomic assays, all cell types and all perturbagens were profiled. But for some other assays that were not so high throughput, only a partial number of combinations of perturbagens and cell types were assessed. And then we had this data coordination integration center, which had 3 sites. One was in the University of Miami, one was in New York, and one was in Cincinnati. And we were tasked for integrating this data and kind of analyzing them, finding some reasonable uses. So links generated a lot of data, but again, I will focus mostly on this one data set that was generated by Broad Institute Transcriptomics Center, and these are the same people that introduced connectivity map concept. This is outdated figure. Now there are 3 million samples profiled at this point, with over 1 million signatures, right, because we have some replicates. And all of these numbers are scale up, but it still gives you the idea of the scale of this project, right? In this stage of the game, which was maybe a year ago, before the latest release, there were 7,500 genetic perturbations that were profiled. That these could be overexpression or genetic loss of function type of perturbations of cell lines, more than 20,000 small molecules across 77 different cell lines. And so we have this huge matrix, right, of transcriptional signatures. Each row is a gene. Ultimately, let's assume that we have 12,000 genes. Story is a little bit more complicated, but I'm not going to get into details of the technology that was used to profile here. So we have readouts on 12,000 genes, right, 12,000 rows, and each column is one of the signatures, one of these 500,000 signatures. Red means that the genes, right, in this particular column have been upregulated by a specific perturbation. and blue means that it was downregulated, black is a neutral, right? Not much has happened. So that's our reference data that we're going to be using in kind of trying to assess this adverse outcome pathway for a toxic. So what are we going to be now doing here is, suppose that we have some kind of perturbation acting on our biological system. This is now represented by one of these pathways, MAPK signaling pathway. And what happens, right, so this perturbation interacts with some of the elements of this pathway, which then kind of triggers a signaling cascade that results in activation of some of these transcription factors and upregulations of or downregulations of genes. And we we get a readout, right, of a state after the perturbation by either measuring here, like these readouts represent either gene expression levels, this heat map, there's a traditional way to do this, or say protein expression level. And again, you know, I mentioned this proteomics because there are some proteomics data that came out of links, and there are some proteomics data that are in iLinks, but most of the time I will be actually considering this gene expression profile. And again, now genes are, you know, each of these bands is a gene, and the red means it's upregulated, blue means it's downregulated. And so it's, you know, if we knew what's happening, right? So for example, if we know that perturbation is a disease-related perturbation that say has constitutively activated RAS, and then in some context that leads to a cancer. And then if we know the mechanisms of different drugs, right, where in this what kind of effect they would have on this signaling pathway, we could maybe reverse the effects of this disease causing perturbation by one of these treatments, right? So RAS would maybe activate RAF, but then some of these drugs would inhibit RAF, right? So we would kind of reverse this aberrant signaling and maybe reverse the disease itself. The problem is that we really don't know what happens, what are these initiating events that cause the diseases, and often we don't have information about what chemical is doing with a specific pathway. So we don't really know, right, based on this readout. But what we would like to kind of learn based on this readout is whether this original perturbation is somehow affecting the signaling cascade, and then also what are the chemicals that could reverse this effect. And ideally, you know, we would want this mechanistically, right, instead of just blindly looking at correlations between gene expression profiles. So, but that is somewhat non-trivial task, right? So if we are to use, so how would we go about figuring out whether, based on the transcriptional signature, whether this pathway is affected or not? The standard process is that we construct this 2x2 table, right? We look at genes that are differentially expressed based on the gene expression profiles, and we look at the list of genes that code for proteins in these pathways, right? And we look for the overlap, right? The genes that are both differentially expressed and are in the pathway, and try to assess the association between differential expression and the pathway by analyzing this 2x2 table, right? So the question is this overlap unusually high, right? And the association would be implied by the fact that the proportion of differentially expressed genes in the pathway is higher than the proportion of differentially expressed genes in outside of the pathway. And this, you know, you do this using a chi-square test. The problem is that in the context of these signaling cascades, the the changed activity of these proteins, right, when there's, for example, they're stimulated or inhibited by a drug, is really not mediated by the changes in their expression, right? So this pathway can be activated, but none of the expression of these genes is changed. So this strategy is not going to work, because when we look for differentially expressed genes, they are not going to be in these pathways. They will be some regulated by these transcription factors, but not necessarily part of this pathway. And therefore, this generally doesn't work for this for pinpointing these early events related to changes in the signaling activity. So how are we gonna go? What is the alternative to that, right? So I will kind of switch to this other small pathway to make this point, and it's kind of prototype, right? And it's related to mTOR signaling. mTOR signaling is hugely important, and it's every day a new biological context is found where it's driving the disease, and you know, there are thousands of clinical trials currently underway for various mTOR inhibitors that kind of treat different conditions. So this is a small signaling cascade, right, in which AKT1, and it's very oversimplified and all this kind of stuff, just to make the point, right? So the, you know, AKT inhibits TSC2, this in turn inhibits Reb, which is activator of mTOR. Ultimately, the activity of the mTOR protein, and these are the genes, corresponding genes, right, names, is gonna determine whether the pathway is activated or not. And the rest of the signaling cascades kind of defines whether the mTOR is going to be active or not. So so this is kind of the prototypical case. Is also what's important about this is that these core proteins, right, mTOR is not really transcriptionally regulated in the cells. Its activity is vastly or almost exclusively in most contexts defined by these by its secondary the phosphorylation status, right? It is kind of these post-translational modifications that are kind of facilitated by these interacting proteins, right? So if mTOR is activated, we will not see change in expression of mTOR genes. We're going to see the increase in phosphorylation of mTOR protein, and the other genes in this that regulate this are also not changed transcriptionally, right? So it's generally very difficult to pinpoint changes in activity of mTOR signaling by using this standard enrichment analysis of gene expression data. So what happens, right, when we treat a system with mTOR inhibitor, which is in this case sirolimus, is that we can observe a transcriptional signature of this inhibition, right? But none of the genes that have changed, and here I'm showing just 6 of them, right, just to kind of have an easy discussion. There are many more that show up, but none of them are actually part of this core signaling cascade. So there is no overlap, right? And actually, if we run, say, in a typical system, right, we treat a cell line with a sirolimus and then run this enrichment analysis. These are the pathways that show up, and none of them really, none of them are basically incorporating these proteins or genes, and they're mostly related to basically proliferation, right? So you inhibit mTOR, you will reduce proliferation perhaps of this cell line, but you will not know why, right? You will not know that this is happening because mTOR signaling has been modified. So the strategy here is that now we look at these genetic perturbations or genetic loss of function signatures for each of the genes in this pathway, right? So for each of these perturbation, we get the signature. So we knock down a gene, and now columns are the signatures for each of the knockdowns, right? So, and what we then do, we can do that for, on a large scale. On links, this is done for like 5,000 genes, right? And we can look at now correlate, right, our knockdown signatures with our sirolimus signatures, and try to figure out and collect, right, knockdown signatures that are unusually highly correlated with the sirolimus signature. And if we now look at the knockouts of signatures of these proteins, right, or genes in this pathway, you see that they are really highly correlated with our sirolimus signature. So if we now, instead of looking at enrichment of the differentially expressed genes, look at the enrichment of genes with the high correlations of their knockdown signatures to our original signatures, and run the same enrichment analysis, right? But now on these genes, we immediately pick up mTOR signaling as the most enriched pathway, or most affected by this treatment. So in this case, this works magically, right? So we go from not having a clue about the origin, the initiating event, right, for sirolimus effect, to basically pinpoint immediately the signaling pathway that is affected by sirolimus. You can go a step further with this and basically use the patterns of this expression, right, or correlations, whether it's positive or negative, combined with the topology of this interaction, right, whether it's inhibitory or activating interaction, and see whether whether they fit. And it's actually, in this case, everything fits perfectly, right? When we have inhibitory interaction between the two nodes, the correlations is negative, highly significant, but negative. And when we have activating interaction, they have a positive correlation, which here is kind of implied by this same color. So we actually, Yan Ran has a paper on this that we wrote on using this topology, right, along with these knockdown signatures to predict signaling pathways affected by bioactive molecules, right, just by looking at their transcriptional signatures. And if we look at, say, mTOR inhibitors, what we do, we look at, across all pathways in, say, KEGG, we look at how well we can, for a given mTOR inhibitor signature, how well we can predict that it's actually really inhibits mTOR pathway versus any other pathway. And it turns out that this switching from actually looking of enrichment of differentially expressed genes to enrichment of genes whose transcriptional signatures of the knockdowns, right, of their knockdowns are correlated with the signature of the perturbagen, increases the precision of the analysis dramatically, right? So this purple line is not using the knockdown signatures. We have two different methods that are not using, right, the signatures, just looking at enrichment of differentially expressed genes. And then we have one line, right, the ROC curve that is actually using signatures, but it's not using this topological information that defines actually direction of the signature that we would expect based on the interaction type of protein-protein interactions. And then finally, the ones that the model that includes both of them actually shows the best results, right? But even this one where we don't really use this additional information about the topology shows a significant ability, right, to detect the mTOR pathway as the target. So the strategy works. And now just to tie it back into our adverse outcome pathways, right? If we have a transcriptional signature of toxicant, we could kind of, by looking at these different types of analysis, we could look ascertain perhaps its mechanism of action by pinpointing these either one or the specific protein that interacts it, or the pathway that interacts with, and then based on the nature of these changed gene expression protein expression levels, we can also infer what kind of changes have been introduced, right? Are the cells proliferating, non-proliferating, and what kind of changes this could cause in the tissue and its function. So that was the purpose of this exercise, right? That we have now a mechanism to maybe look at or method to look at these mechanisms. And the utility here is that transcriptional signatures of a toxicant is really relatively easy to generate, right? And again, there is an additional kind of layer here in addition to these mechanisms, right? Because we could, once we know the mechanism, we could compare this to the mechanisms of the toxicants with the known adverse outcome pathway, in which we know the mechanism and we know the adverse outcome. So if we can match the mechanism of a new toxicant, we can kind of at least hypothesize what adverse outcomes ultimately can be for the toxin. All right, and now this leads us to iLinks, right? So iLinks is meant to allow you to do all of this stuff really easily by just pointing and clicking using a single mouse button, right? You don't even need the right click, although it sometimes helps. And the portal it consists of user interfaces and analytical engine, and then databases of pre-processed omics data sets, actually proteomics and transcriptomics, although most of them are still transcriptomics. There are now more than 12,000 actually data sets in the backend. And then these signatures that I was talking about, we have these link signatures in addition to many other signatures that some of them I will mention later. Total of 222,000 signatures. Again, we don't use all link signatures. They're not in the backend because we have some QC measures first to filter out signatures that don't seem to be informative or did not they're not strong enough to be used in the analysis. We end up with about 200,000 link signatures. There are signatures profiling effects of more than 20,000 small molecules, and this is obviously highly inaccurate. It's 74,000 because we actually have data on mouse, rat, and humans, right? So each of the same gene in different species is counted separately. So and I would like to acknowledge the lead people on this project are Marcin, Michal, and Becruz. Marcin has designed the user interfaces and analytics. Michal has designed the backend infrastructure, which is pretty complex here. Consists of the load balanced Docker containers, right, in a swarm that can perform a lot of these pretty heavy duty analysis for multiple queries at the same time, right, and it doesn't get overwhelmed and so on. And then Becruz has taken over recently development of UIs and also the backend analytics. So how does this work, right? So the idea of analysis through iLinks is that you pick up a signature, and this signature can be one of these 220,000 pre-computed signatures, which again, a signature consists of differential expression levels after perturbation compared to the control. And we also provide associated p-values, but these changes are really the core signatures. So we have 220,000 of these. or user can submit their own signature, right? They create the signature using their own experimental data, submit it to iLinks. Or the third way is to use, oh, okay, so now this number is 14,000. So we have more than 14,000 data sets, right? Or they can use one of these 14,000 data sets to create a signature, right? So here you don't even have to run your own experiments. You just query this large collection of data sets that are already there and analyze it and create. An analysis is like three steps by pointing and clicking, right? Selecting the data set, selecting samples that you want to compare, and clicking submit button. So ultimately we end up with the signature, right? Now the analysis of these signatures, and these are all implemented, right, in iLinks, and you kind of go from, once you create the signature, to one of these analyses of these signatures by just, again, pointing and clicking. And there are methods that implement this pathway analysis, enrichment analysis, and network analysis, and they also iLinks leverages outside tools, right? So it's basically gonna submit your signature directly to one of these tools and redirect you to these tools that are sometimes really optimized for this particular type of analysis. But then the other path that you can undertake, and that's what I was talking about most of the time so far, is that you don't really look necessarily only on differentially expressed genes directly, but you connect them first, right? You connect your signature to this large collection of perturbation signatures, and then in addition to this baseline analysis, you can analyze the results of these correlations, right, with pre-computed signatures, and in some cases, as I demonstrated with this mTOR pathway, right, this can offer dramatically more improved or dramatically improved precision of your analysis. So ultimately, this can lead to biological insight targets and adverse outcome pathways in this case. So just to give you a little bit more on the signatures that are there, right? We have again signatures of genetic perturbation of genes. So these are either knockdown signatures, these consensus G signatures, 37,000 of them, overexpression signatures, and then chemical perturbation signatures, which have all drugs, but also other bioactive small molecules that we don't often have no idea what they do, right? But they produce some kind of transcriptional response. We also have targeted proteomic signatures, which I'm not going to discuss here. And then we have a bunch of signatures that are non-link signatures. They are derived from either other large-scale projects, like, for example, transcriptional signatures from EBI Expression Atlas, or we constructed them by analyzing systematically, say, geodata. These are these disease-related signatures. So all of them can give you, in some context, some information when you compare your signature of interest to them, and I will show a few of them how they work. And then in terms of data sets, right, we have these links data sets, right, that are not that many of them, actually 15, but some of them are really big. We have full collection of TCGA. We have this GDS collection actually has all the Geo 3600 Geo data sets that are like curated Geo data sets. We have the other one that is really interesting is this grain collection that has basically a large fraction of all RNA-seq data sets from Geo pre-processed. So you don't need to do anything. You just go there, select your data set, select your samples to compare, and click submit, and it creates the signature, right? It runs differential gene expression analysis for you. This number is much bigger now, and the way this portal works, we processed a whole bunch of this on an initial initially, but now there is the submission tool, right? So if we didn't process the signature that you are interested in, dataset that you're interested in, all you need to do is submit its geo identifier, and it's going to be put in the pipeline and processed, and it's going to be most situation it's going to be ready in 24 hours or 48 hours for the analysis. So basically you can say that you can access any RNA-seq dataset in geo using this mechanism. All right, and then, in terms of analysis and interactive visualizations, anything that you can think of in terms of in this context, right? We have interactive heat maps, interactive volcano plots, TSNEs, PCA plots, 3Ds, 2Ds, GSEA plots, depending on the context, right? Different visualizations are provided, and again, we link to a bunch of tools, right? So once you create the signature, you can submit to some of these tools directly and run the analysis that they are best at. So finally, an example, right, I'm sorry. So first demonstration of this mTOR thing, right? So that I was talking about before. So we query our signatures. We pick up, say, a everolimus signature with concentration of 0.04 micromolar and 24-hour duration of treatment in MCF7 cell line. And try to figure out now, pretend that we don't know what everolimus does. Actually, we know it's mTOR inhibitor. But again, the results I showed you before, if we just run enrichment analysis on this signature, we don't get anything mTOR related. But now we first run connectivity analysis against these consensus genes knockdown signatures, and then instead of running enrichment on differentially expressed genes, we run enrichment of correlated genes, right? The genes that produce signature, knockdown signature that's correlated with our query signature, and run enrichment analysis of these, and again, the same mechanism, right? Enrichment analysis mechanism now identifies mTOR as the pathway of interest. And now how an application, say, how we would perhaps use one of the data sets that's already there, pre-processed for you for assessment of adverse outcome pathways for Printex 90, right? These are nanomaterials, and so the experiment is a mouse treatment by inhalation by this Printex 90, and then lung transcriptional profiling after treatment, right? And so we use this data set, we go through this create signature workflow. It just tells us, okay, select your samples to compare, and we compared samples treatment with 162 micrograms to no treatment, right? And the number of samples in each groups was 6 and 5, right? And the constructed list of differentially expressed genes. And now we run this connectivity analysis, and we use only upregulated genes for the first for one of these analyses, and we get this fairly expected. This in the original pathway, inflammation was identified as the original article, right, as the effect of this treatment, and we kind of identified the same thing, right? We got identified TNF signaling, for example, as one of the pathways, and then we can map again on iLinks, right? Allows us to map these genes that were upregulated, right, to this pathway, and most of these are actually downstream, right, from the pathway, meaning they're downstream from this activated transcriptional regulators, right? So that makes sense because these genes are upregulated after activation of this pathway. But then the next analysis we do, again, now we connect first over these upregulated genes with overexpression genetic perturbations, right? In this case, we don't look at knockdowns, we looked at overexpression assays or perturbations, and now run basically enrichment analysis, again, pathway analysis on the genes that were targeted, right, that were overexpressed and produced correlated transcriptional signatures. And actually similar things show up again, right? Our TNF signaling is still significant, but and some other related pathways show up as well. But just to kind of but again, I will look at this map now. TNF related genes, right? That showed up as significant here, and now these are kind of there seems to be a similar type of output, but it's really fundamentally different, because in the previous one we kind of look at the consequence of this treatment. Here we are looking at alternative perturbations, right, or that lead to the same type of biology, right, same type of transcriptional signature. So these so overexpression of or activation, you can think of activation or overexpression of any of these genes or proteins actually can lead to the similar type of effect that we observed by treatment with these nanomaterials, which kind of gives you the more of a mechanistic interpretation really of these signatures, right? That truly this pathway, activation of this pathway or is leading to the changes that we observed. We can combine these results, right, to kind of see what genes are come out as a transcriptional consequence of activation of the pathway versus the genes that actually can activate the pathway and lead to this kind of transcriptional program. We can also compare this to, say, chemical perturbagens that potentially could reverse the effect, right? So now we would look at negative correlations. And again, one of the pathways that shows up, right? So for each chemical perturbagen for which we know the target, we can now run enrichment of the targets, right? And identify that maybe inhibition of MAPK signaling, right? And that can lead to maybe decreased of inflammation, right? Which makes perfect sense. So this is about the mechanism, right? So these first two boxes of adverse outcome pathway model, but then we would maybe also like to connect, if we didn't know what this Printex 90, right, this nanomaterial ultimately lead to in terms of diseases, we run one of the things that maybe hypothesize at least what happens is to run the comparison with the with the disease signatures, or in this case signatures of other perturbations, right, that are not necessarily have not been part of the LINKS project. So these are signatures again picked up from the EBI expression atlas. And if you look at these most correlated, positively correlated signatures here and how they are created, we can see that they are all related to some kind of inflammation induction, right? And we have, for example, effect of FOS gene, right, in the mouse lungs, and these are all actually turned out to be in mouse lungs experiments, right? So it turns out that the effects of the FOS gene is similar in this acute effect as the in terms of transcriptional signature as the effect of this nanomaterial. And so this kind of gives you now at least a hypothesis that if we know what FOS gene does long term to mouse, right, given this acute inflammation that it induces, we can actually hypothesize that this nanomaterial is going to do the same thing long term, right? So that kind of gets us to perhaps the outcome, adverse outcome. Finally, I would just want to mention that if you want to interact with iLinks programmatically, you can do that as well, right? So you can include these calls in your R script, and we have an example of R scripts that does this programmatically. Most people interact with it using its user interfaces, and I think this is the biggest strength of iLinks. And I would like to acknowledge more people. So I acknowledged 3 main leaders of the iLinks project, but other people that were involved, right, and contributed. Jarek is a co-conspirator in both TCIC and CEG Bioinformatics. John helped a lot during this development of iLinks and in many other. He was part of our DCIC team. Siva is our real Bayesian statistician in the math department who helped with the modeling a lot. Jacek is a research associate in CEG. Jan Ren is a postdoc who again contributed with this methodological research. Wen is an application developer, and then Kurt, you all know probably, and then students and postdocs that participated to some extent in the development of the portal. Avi Mayan and Stefan Schurer are PIs of 2 other sites of DCIC. They also contributed to the development, and our LINKS NIH administrator were really helpful during these 5 years of working on this project. Again, work has been supported by DCIC and CEG. Thank you for your attention.